This year's Nobel Prize in Chemistry is about tailoring proteins for the benefits of humans and our planet. Proteins are our best friends. They are everywhere inside us. They help us move and see and think. To the left, you see an enzyme. That is a protein that can speed up chemical reactions, a biocatalyst. To the right, you see an antibody. That is a protein that can grab onto foreign objects and protect us from invaders. This year's laureates harnessed the power of evolution to make even better proteins that can be used to catalyze chemical reactions in a sustainable way and to treat diseases. They developed methods that combine advanced knowledge about how proteins work with a bit of good luck to be able to find improved variants beyond what we may guess. That's a very smart trick. It takes the best of what we know and asks the experiment to tell us even more. This year's Nobel Prize in Chemistry unifies two aspects of protein evolution. One half goes to Francis Arnold for the directed evolution of enzymes, and one half goes to George Smith and Gregory Winter for the phage display of peptides and antibodies. Here we will now first learn about phage display. George Smith was born 1941 in Norwalk, United States. For his college education, he took a very wise decision and went to Haverford College, a warm and nice liberal arts college in Pennsylvania. He then went to Harvard for his PhD in Cambridge, United States, the other Cambridge, as we call it in Europe. He's now an emeritus professor at the University of Missouri. George Smith developed phage display. This is an ingenious method that provides a physical link between genotype and phenotype, what's inside and outside. The method has become extremely useful for deriving knowledge about binding determinants, to derive better binding proteins, and also to improve proteins in many other ways, including stability. George Smith, I now welcome you onto stage. We are very much looking forward to hearing your lecture.
When this award was announced on October 3, my friends who were actual chemists were surprised to learn that I was one too. And that brings up the question of what is a chemist? Of course, we might try to use anecdotal evidence to recognize chemists by their behavior. For example, two chemists are toiling away in the lab when a funeral procession approaches on the road below, and chemist one stands at the window with his hand over his heart until the funeral procession has passed. And afterward, chemist two says, "I am in awe of the reverence that you show to the dead." And chemist one says, "Well, she was a darn good wife." My darn good wife, Margie Sable, is very much alive and sitting in the audience. As are our darn good sons, our darn good daughter-in-law, my darn good brother and sister-in-law, my darn good sister and brother-in-law, and some darn good colleagues, some of whom will figure in this lecture because they were directly involved in the experiments that I'm about to describe. And by the way, in case you have seen the movie The Wife, I would like to assure you that Margie didn't invent phage display. Actually, I think it may be a little doubtful that phage display was an invention. I know that it looks like that from the outside, but from the inside, from my point of view, phage display was a series of small incremental advances from the knowledge that I had, the ideas, the published findings, and so on, that my science community had made available to me, and that are the nutrients of my science community. So my science community is the global overlapping community of molecular biologists, of evolutionary biologists, of immunologists, protein chemists, phage biologists, etc. So I feel that I'm accepting this award as a representative of my science community. And I say my science community because I belong to the community, not because the community belongs to me. And so perhaps a better way is to say that it's our science community. And it's our science community in a broader sense as well, because the society as a whole does, in so many myriad ways, sustains our science communities, including mine. The schools educate our children, some of whom go on to be professional members of science communities. They go to our universities for their continuing education, including my university, the University of Missouri, that not only educate our children, but also sustain our local science communities, and also our national institutions, such as the National Academy of Sciences, the National Science Foundation, the National Institutes of Health in the United States, the Medical Research Council in Great Britain, that promote science, that do some science themselves, and that support science fiscally. So let me introduce phages. Phages are viruses that infect bacteria. This is the phage, the class of phage that phage display was developed with. They're called filamentous phage because of their shape, long and thin like a filament. Phages in general have many different shapes, but they have this in common, that they have an outer coat of protein made up of phage coat proteins. And that coat surrounds or encases the phage genome, the phage chromosome that has the phage genes. And the particles are about 1 micrometer long. That's about half the length of the cells they infect, but only 6 nanometers in diameter. So they are extremely thin compared to their length. I've mentioned chromosomes, genes, and proteins, and let's just be clear, because I'm going to give you a crash course in molecular biology. Let's be clear the relationship among them. So the DNA chromosome is a long chain of the 4 nucleotides, A, G, C, and T, in a specific order or sequence. And a gene is a segment of that chromosome that specifies or encodes, as we say it, a polypeptide. So a polypeptide is a long chain of the 20 natural amino acids, again, in a specific order or sequence that is specified by the gene. And polypeptides in general will fold spontaneously into their natural 3-dimensional shape. That's the native protein that has the protein's biological activity. I would like to introduce one more term that will appear, and that's peptide. So a peptide is a short chain of amino acids. And in the context of this lecture, peptides are encoded by short segments of DNA in genes. Back to phage, I would like to briefly outline the phage infection cycle, very simplified. And I'm drawing the phage about 3 times bigger than its size relative to the bacterial cell that it infects. So the first stage is attachment of the phage to the cell that it's going to affect. And within minutes, the phage chromosome is intruded into the interior of the cell without killing the cell. And over the course of the next hour or so, the phage chromosome replicates, so now there's many chromosomes inside the cell, and the phage proteins that are specified by the phage genes, including the coat proteins, accumulate in the cell. And ultimately, the cell extrudes progeny viruses, phages in great numbers, perhaps 100 within the first hour. Filamentous phage are a little unusual among phage in general in that they don't kill the cell they infect. So the infected cell continues to grow and divide, although at a lower rate than uninfected cells, and continues to extrude progeny phage. Meanwhile, these progeny phage are able to infect any uninfected cells in the culture, and so soon all the cells are infected. So let me outline what this means in numbers. So if we start with a flask with 1 liter of culture and about, say, 10 to the 11th cells and 1 phage particle, and shake that vigorously overnight, we end up the next day with about 50 times more cells and about 10 to the 15th phages. So 10 to the 15th, some of you may not be familiar with that number. So 10 to the 6 is a million, 10 to the 9 is a billion, 10 to the 12th is a trillion, and 10 to the 15th is 1,000 trillion or a gazillion. And that's and this is a tremendous advantage of phages as a study organism, because they can be produced in enormous numbers very cheaply and very simply. So phage display. So I've done with phage now, and we're getting on to phage display. Phage display had its origin at the end of my sabbatical leave with Bob Webster in the biochemistry department at Duke University. This is a picture of Bob recently cleaning up after Hurricane Florence earlier this year at his retirement home on the North Carolina coast. But he was a major investigator of the biology of filamentous phage. And I was a newcomer to the field, and that's why I came to his lab for my sabbatical. And much of my research was focused on a particular coat protein gene. The coat protein encoded by that coat protein gene was at the tip of the virus, the tip of the phage particle. It's a multifunctional protein. Part of it is buried in the particle and is essential to the structural integrity of the particle. But the rest of it is exposed out to the surrounding medium and plays critical roles in the infection process. And it was also known through many channels that this protein was the exposed parts were flexible, that they could be manipulated, that they didn't have a set, tight structure. And that suggested to me at the time that it might be possible to insert foreign pieces of DNA into this coat protein gene and into the part of it that coded for the parts of the protein that are exposed on the surface. And as a result, the extra peptide, it would make a longer gene encoding not only the regular coat protein, but also a little extra piece. And that extra piece, the peptide that extra piece corresponds to, would be displayed on the surface. So down the hall from Bob Webster's lab was Paul Modrich's lab. Here he is getting his chemistry Nobel Prize 3 years ago. And he was studying a protein called EcoR1 at the time. And he gave me the gene for EcoR1 and antibody against the EcoR1 protein. So here's what I did with it. I cut out a piece of the EcoR1 gene and inserted it into that coat protein gene. And the expectation was that the resulting construct, the resulting modified phage, would have a piece of the EcoR1 protein, a peptide from EcoR1 protein encoded by that piece of EcoR1 gene on its surface at the tip. So this is the first phage display construct. And I should say that from now on in the lecture, I'm going to simplify the cartoon representing phage display constructs. So I'm going to eliminate representing the chromosome and the host coprotein gene and just have a colored rectangle representing the foreign DNA insert that's in that coprotein gene. Well, I told you that Paul Modrich gave me the antibody against that protein. So I should say that this construct, this phage, was still infective, but when I mixed it with the antibody against EcoR1 protein, it blocked infection, presumably because it bound to that piece of EcoR1 protein on the surface and prevented infection. And this was the first indication that the phage display concept seemed to be feasible. It seemed that it was possible to display foreign proteins or pieces of proteins, peptides, on the surface of phage. I was very excited about this, and it's very likely that many of you don't understand what was interesting about that. And so please wait. I will try to make it clear. But I was really interested. So when a week or so later, when Margie and I drove back home to Missouri, I soon swung my research program over to developing the phage display idea. And luckily, a darn good colleague, Steve Parmley, showed up as a graduate student in the lab. And Steve had two projects. One project was to develop a really practical phage display vector, a vector that would make it easy to insert foreign pieces of DNA into the coat protein gene, having minimal effect on the function of the phage, so it would still be infective, still be able to be produced. And that wasn't an easy project. He tried many, there were many tries, some of which didn't work out so well. So one of them was Pig3C. Pig3C didn't work too well. At least it certainly wasn't the best construct, but it soon became a mascot in the lab. And Steve is going to take this over to the Nobel Museum as one of our artifacts. The second task that Steve took on was developing affinity selection. And I know those words don't mean anything to most people in this audience, and that will be explained presently. So just as Steve left the lab, another darn good colleague, Jamie Scott, came as a postdoc. And Jamie Scott was the first one to make a large random peptide library, and I'm going to describe that in a minute too, and to use Steve's affinity selection process, also to be described presently, to select peptides from that library. And I would just like to mention a third darn good colleague, that's Robert Davis. Robert Davis came to my lab just about the same time as Jamie, as the chief manager and technician. And he was a wonderful and wonderfully productive technician with just marvelous intuitive lab sense. And he had many tasks, and one of them was that he was our DNA core. So what it meant in those days, that he was responsible for all the DNA sequencing in our lab. We calculate conservatively that he sequenced 1 million bases of DNA using the old-fashioned radioactive technology. And if you've ever used that, some of you are old enough to have used that, you know how laborious that is and how technically demanding it is. So I hope Jamie will forgive me for not using the work that she published in 1990 as the illustration of random peptide libraries and affinity selection. I'm going to use a work that involved a model system that came to our lab a few years after she left, because I think it's easier to explain. So this was brought, this was the S protein system that was brought to our lab by two graduate students from Yale, John Ladbury and David Schultz. And it's the S protein system. So the S protein system derives from research of Fred Richards at Yale in the late 1950s. He was studying the structure of the enzyme ribonuclease, which is represented here in three-dimensional form. And he showed that the proteolytic enzyme subtlysin would split ribonuclease into two pieces. One piece was the S peptide that was only 20 amino acids long, and the other piece was the S protein, which is 104 amino acids long. And those two pieces could be separated from each other, and when you did that, neither piece had the enzyme activity of ribonuclease, but when you added S peptide back to S protein, it bound to S protein and restored enzyme activity. This is the S protein system. And we regarded it, S protein, as kind of like a stand-in for a receptor. Maybe a receptor involved in a disease or something. It's kind of like a pretend receptor, because it was we could even buy it commercially. And maybe we could think of S peptide as the natural ligand for that receptor, say a hormone or something. And maybe when that hormone is overproduced and it binds to the receptor, you get the dreaded S protein disease. And let me assure you, this is fake news. This is just pretend. But this is the way we thought of this as a model system. So I'm going to introduce random peptide libraries and affinity selection in the context of an experiment that was done in the middle 90s by Jinan Yu in my laboratory, as a graduate student in my laboratory. So she had this S protein receptor, which you could get in pure form. And she also had a random peptide library. So let me describe what that is. A random peptide library is a huge collection of phage clones. And each clone differs with regard to the, foreign DNA insert that's inserted into the coat protein. And these are short DNA inserts, and the particular library that she used had inserts that were long enough to encode 15 amino acids. And these were random DNA sequences. And as a result, random 15 amino acid peptides are displayed on the surface of the phage at the tip. Round blue peptides, sharp gold peptides, square green peptides, etc. And actually, her library had 250 million different clones. And I assure you that the random DNA inserts didn't have to be synthesized one at a time. That would be an impossible task. They could all be synthesized together in a single vesicle in one operation. And also, they didn't have to be inserted into the coat protein gene one by one. That all those insertions could be, again, be accomplished in a single vessel in a single operation. So here's a picture of the library. In that bottle, there are 10 to the 15th phages, representing 250 million different phage clones, each one displaying a different random 15 amino acid peptide. So here was her task. Her task was to find, out of that 250 million peptides, very rare peptides that would happen to have the ability to bind to that receptor, to that S protein receptor. You can see this would be a really hard task. How are you going to go through 250 million phages one at a time, especially if they're all mixed together? But you didn't have to do that because of the affinity selection procedure that Steve had previously worked out. So let me describe affinity selection. First, the receptor was immobilized on a solid surface. It was the plastic surface of an ordinary plastic Petri dish. Next, the library, or a portion of it representing every one of the clones many times over, was poured into the Petri dish and sloshed around for an hour or so. And during that sloshing around period, if there was a peptide that had good affinity for the immobilized receptor, the peptide and the phage that it was attached to would be captured on the surface of the Petri dish, whereas all the other phages, the vast majority in the library, would be floating free. And that allowed those floating free, those unbound phage particles to be washed away, leaving just the particles you want, and they could be released, and you're home free, but you're not, because the released particles were released in tiny, tiny numbers. So there are two reasons for that. One is that any phage clone that happened to display a peptide with high affinity for the receptor would be a tiny minority of all the original phage clones there. And furthermore, the capture process is not very far from 100% efficient, maybe more like 1% efficient if you're lucky. But it doesn't matter, because remember that these particles are infectious. Simply by infecting fresh bacterial cells, you can produce them, you can amplify them up to trillions or gazillions. So this is the peptide, using the one-letter abbreviation for the amino acids that Jinan selected in her experiment. Although it's very important to point out that never did we use any information about S-peptide in either the synthesis of the library, the construction of the library, which was done by collaborators in Japan, who made the library available to us, and certainly didn't have S-peptide or S-protein in mind, and nor was it used in affinity selection. Nevertheless, I think it's instructive to compare the sequence of the selected peptide with the sequence of S-protein, the so-called natural ligand. Well, they match in 5 positions, and 4 of them are very important, the ones in red. Because that happens to be matches in precisely the 4 amino acids of S-peptide that were known structurally to be deeply buried in S-protein when S-peptide binds S-protein. So these are the residues of S-peptide that are responsible for the tight binding, and the selected peptide matches them. So in a certain sense, this is really kind of a successful experiment. And I think I've explained enough about it. I just want to make a generalization that this artificial selection in the Petri dish mimics, in some important respects, natural evolution in the living world. Natural evolution depends critically on diversification, generating varieties of variants of living things. And in phage display, what corresponds to that is making enormous phage display libraries with great diversity of peptides displayed. Secondly, evolution depends on natural selection to favor some variants and disfavor others. And so, too, in phage display, affinity selection greatly favors some peptides over others on the basis of their affinity. So this is a very simplified kind of selection that the experimenter controls and decides for the experimenter's own needs. And this diversification and natural selection is the engine of adaptation in the natural world, evolution in the living world. And so generation of great diversity and affinity selection leads to peptides with desired activities. So let me summarize. This again is the library, and by applying her specific selection protocol to that library, she was able to discover in that library a cure for the imaginary S-protein disease. But what end would there be to other marvels that could be selected from the same library using other selection protocols. Marvel 1, Marvel 2, who knows what end there is to this. And indeed, several other members of our science community have filled bottles like this, numerous bottles like this, with other phage display libraries of novel and imaginative design. And from those libraries, some remarkable marvels have been selected, some of which you'll hear about from one of my co-laureates and our next speaker, Greg Winter. Thank you very much.
Sir Gregory P. Winter was born 1951 in Leicester, United Kingdom. He got his PhD 1976 at the University of Cambridge, the first Cambridge. And he's now a research leader emeritus at the MRC Laboratory of Molecular Biology in Cambridge. He realized early on that phage display could become a very useful method when you work with antibodies. This would be a tool to derive very high affinity antibodies for any target that you wish. With strong determination, he spent all the effort required to make it possible to display antibodies or antibody fragments in functional form on the surface of the filamentous phage, so that he could then fish out the good members from libraries. Sir Gregory Winter, I now welcome you on stage.
Thank you. Thank you. Well, good morning, ladies and gentlemen. It's still just the morning. Sarah, thank you for your introduction. May I also thank you and your colleagues on the chemistry committee and their advisors for all the hard work they had to do that ended up with this, with me getting a share in this prize. So thanks. I would first like to acknowledge and thank my family. It gives me great pleasure they're all here today and will have to listen to me for a whole half an hour without interrupting me or saying, "Dad, we didn't want a lecture on it." So I would also like to thank the academic institutions that oh dear, this is going to start messing about. To thank the various academic institutions who've supported me throughout my scientific life. In particular, the MRC, the Medical Research Council's Laboratory of Molecular Biology in Cambridge, the Medical Research Council Centre for Protein Engineering in Cambridge, and Trinity College, Cambridge, all the same Cambridges. I would also like to acknowledge the key role of a startup company, Cambridge Antibody Technology, fourth time I've mentioned Cambridge, in the work that I'm going to describe. I'd also like to thank some individuals. I hope they recognize themselves. From some years back. I'd particularly like to thank John McCafferty, who was the first to show the display of functional antibody fragments on phage. I'd also like to thank Alan Furst for his hand in a number of things, but particularly when he was director of the Centre for Protein Engineering for accommodating the greater part of my group as it was developing the phage technology. And all the others on the slide, who are probably the most important because they did the work. They did the work at different times, they all had key roles, and they are the anonymous we that I shall refer to during the talk. Finally, I would like to acknowledge Richard Lerner and his group at the Scripps Research Institute. although we were certainly scientific competitors at that time, their activities complemented our own and actually confirmed our own faith in our work. It actually helped us persevere through many technical difficulties, because if we knew that if we didn't manage to do it, they would. So first of all, you've had a tutorial on phages. I'm now going to give you a tutorial on antibodies. Antibodies are part of our immune defense system. They defend us against infectious agents such as viruses and bacteria. They're raised by the immune system in response to infection or vaccination. An IgG antibody, as shown on this slide, is a large Y-shaped molecule. It's got 2 arms and a stem. It comprises 2 heavy and 2 light chains of linked protein domains. In this case, it's the white and the red. The heavy and light chain variable domains, which are at the end of the arms, come together to form a protein scaffold of beta sheet, which I've marked here. That's surmounted by 6 loops, these red loops numbered 1 to 6, which are responsible for binding to the antigen, and in binding to the antigen, they can block the process of infection. In addition, the antibody stem here is able to recruit the immune system to kill the infectious agent to which it's attached. Now, the variability of these antigen binding loops is created through rearrangement of different germline segments of DNA during B-cell development. In heavy chains, it's created through random combinations of 3 segments, the so-called VH, D, and J segments, as marked on the slide, each of which has multiple members. So you get tremendous combinatorial diversity by throwing these germline segments together. In the light chain, it's created by random combination of 2 sets of gene segments. In addition, where they join, there's editing takes place, and further diversity can occur during the DNA rearrangement process, as well as random pairing between the heavy and light chain domains as the heavy and light chains are rearranged independently. So after DNA rearrangement, each B cell, which I've got here, so this is before rearrangement, after rearrangement, it encodes a unique antibody on its surface. So the rearrangement process leads to expression of antibody on the surface of the B cell. As the DNA rearrangement is proceeding in parallel and independently in many B cells, this creates a library of B cells, each with a different antibody on its surface. If the displayed antibody encounters an antigen, the B cell is stimulated to differentiate. And it can do 2 things. It can differentiate to become a plasma cell, which essentially is a factory for production of soluble antibody against the cognate antigen that antibody has recognized. Alternatively, it can become a memory cell, as marked here, where the antibody genes are subjected to random mutation within the cell. On further encounter with the antigen, the memory B cells compete for antigen, and those displaying mutant antibodies, which have got the highest binding affinity, are favored for further rounds of differentiation. This leads to an antibody response that improves with re-stimulation with antigen. It's a process known as affinity maturation. So in summary, the immune system is a simple evolutionary system. At its heart is the B cell. It's a phenotype, a genotype, firmly linked. It's essentially a genetic display package with the antibody displayed on the outside of the cell to encounter antigen and the genes encoding the antibody within. Now, although nature developed antibodies to protect against infectious disease, man has further developed and evolved them for the treatment of non-infectious disease, such as inflammatory disorders and cancer. Man-made antibodies can be used, for example, to block the biology of protein receptors and ligands, such as are involved in inflammatory disorders or cell growth or even T-cell activation, or they can be used to kill target cells by recruiting the immune system to attack. It's the development of antibodies for treatment of non-infectious disease that's revolutionized the pharmaceutical industry. And what this slide shows is the top-selling drugs in 2016. Previously, the pharmaceutical industry was dominated by chemicals, but what we can see here, I've marked in red among those top 10, those antibody products. So actually, the pharmaceutical industry in the top-selling products is now dominated by antibodies. We've got 6 antibodies, and the number 1 antibody is an antibody called Humira, which I will come back to later. These antibodies are mainly used for treatment of autoimmune inflammatory diseases and cancer. The technologies to make such antibody pharmaceuticals required several years of inventions and developments. It started with the invention of hybridomas by Cesar Milstein, actually also at the MRC Laboratory of Molecular Biology in Cambridge in 1995. They immunized mice with target antigens such as human cancer cells. The spleens were harvested, and the responding B cells were immortalized by cell fusion. The hybrid cells, or hybridomas, were then screened to identify those making monoclonal antibodies against the target antigens. Although this technology generated many useful research reagents against human proteins and cells, the mouse monoclonal antibodies were seen as foreign, and when injected into patients, this compromised the use. In fact, it could lead to life-threatening reactions. At that time, attempts to make human monoclonal antibodies against cancer cells proved impossible, not least because the immune system has tolerance mechanisms that prevent it making antibodies against self. Generally, this is fortunate. Where these mechanisms break down, it generates terrible autoimmune disease. By the mid-1990s, solutions for making antibody therapeutics began to emerge through the application of protein engineering. This is a branch of genetic engineering in which the genes of a protein are altered and the encoded proteins are expressed in a host cell. The first solutions here involved creating what we call simple chimeric antibodies, in which we take the entire antigen-binding regions at the end of the mouse monoclonal antibody, that, for example, could be against a cancer cell, and we transplant the entire region into a human antibody. So the human antibody is marked white. So this antibody is 2/3 human, 1/3 mouse, and should therefore perhaps be better tolerated when put into patients. Then came humanized antibodies down here, in which the antigen binding loops were grafted from the mouse scaffold, this beta sheet scaffold with the 6 loops, were grafted from the scaffold to its human counterpart. Now, as the inventors of humanized antibodies, which are up to 95% human in origin, we argued that such antibodies might in practice be regarded as synthetic human antibodies, as in any case, these loop regions differ between one human antibody to the next. Experience has actually borne this out. Humanized antibodies are well tolerated in the clinic and account for the majority of antibody pharmaceuticals on the market. But at the time, we weren't actually so sure that the human immune system wouldn't somehow sniff out the taint of mouse that's in those loops, and it might treat them as foreign. You know, I have a sort of very anthropomorphic view of molecules. I think you need to think about them like that. And I was very uneasy about it, despite my logical arguments that they were just the same as human antibodies. So we were very receptive to an opportunity to make fully human antibodies that emerged from a technical advance. One of the rate-limiting steps in making chimeric antibodies, these are the 1/3 human, 2/3 mouse, was the isolation from the hybridomas of the genes encoding the antibody variable regions. In theory, this is straightforward. In practice, actually, it's a pain in the neck. And to simplify matters, we thought we might be able to use the polymerase chain reaction to amplify and clone the genes from the hybridomas. Now, in the polymerase chain reaction, target regions of DNA, so because I've marked one here, can be amplified by repeated cycles of extension of 2 flanking primers by a DNA polymerase. And by repeating the cycles, one can get a dramatic amplification of that DNA and clone it. But since we didn't know what the sequences of the genes were in any particular hybridoma, it wasn't clear that we could design PCR primers that would work on any hybridoma. What we did was to compare lots of different antibody sequences, and we spotted regions at the ends of, so if we look here, this is a heavy chain sequences. We look at nucleotide conservation at the end. We can see regions which are 100% conserved with regions where they're not conserved. And we thought that maybe by playing around with experimental conditions, we might be able to find a set of PCR primers that could amplify any mouse V gene from a hybridoma. So we set ourselves that task. It involved a lot of fiddling about. It was the early days of PCR. But in fact, we did find a set of primers that worked for most hybridomas. We could actually amplify the genes from most hybridomas we were given. And we later developed a similar set of PCR primers for amplifying human antibody genes. Now, these primers made it so much easier to clone antibody V genes, and it is that presented the new opportunities. For example, instead of isolating the mouse V genes from antibodies from hybridomas, why not amplify them directly from the spleen of an immunized mouse? If we express the encoded antibodies, perhaps we could find the ones with antigen binding activities. This offered a simple alternative to hybridoma technology for making mouse monoclonal antibodies. But better still, if we could make libraries of human antibodies, we might be able to bypass immunization and find ones with binding activities to human self-antigens as we need for therapeutic use. So we were excited by the potential, but first we had to find an expression host. We looked at bacteria. It had recently been shown that functional antibody fragments could be expressed in the bacterial periplasm. We took libraries of antibody fragments from, we took libraries of antibody genes and expressed these as fragments in bacteria. Those genes came from immunized mice, and we soon discovered fragments with antigen binding activity. Now, by this time, we'd become aware of a similar approach being explored by the group of Richard Lerner at the Scripps Research Institute, who had similar results. But both of our groups were completely unable to detect antigen binding without the use of immunization. So clearly, our screening methods weren't really powerful enough, certainly if we wanted to make human antibodies. We therefore looked back at how the immune system manages to select antibodies. As I explained earlier, at its heart is the B cell. Could we find a B cell mimic? We thought of many possibilities: display on mammalian cells, display on bacteria, or even display on bacterial viruses. But it looked like I would need some more staff, but I didn't have the money or the space to accommodate them. At that time, I met David Chiswell and John McCafferty from Amersham International, who, due to an error in the counting processes, were in the process of being made redundant. They were excited about the possibility, actually any job really, they were excited about the possibility, but they were particularly excited by the potential of the library technology, and we wondered about forming a startup company to do that. But it was a struggle to get funding, but finally I succeeded through an old friend, Geoffrey Grigg. He'd set up a startup company, Peptech, in Australia, that was mainly backed by the mums and dads of Australia, but it also seemed to be backed by I would call the horse racing fraternity. I had to go and meet the shareholders on a yacht in Sydney Harbour. And I remember one of them saying, "Oh, I'll give Greg the money. Let's see how the boffin trots," which was... So Peptech, in fact, in the end, became the major shareholder in Cambridge antibody technology. David Chiswell and John McCafferty joined, and John came to work in the Laboratory of Molecular Biology, and we decided to focus our energies on the beast you've heard about in the previous talk, the filamentous bacteriophage. So some 4 years earlier, George Smith had shown that filamentous bacteriophage was capable of displaying peptides, and that the phages bearing the peptide epitopes on the phage gene 3 protein could be selected by binding to cognate antibodies. So could this phage serve as our mimic of the B cell? Instead of peptides, could we display folded antibody fragments on the phage? And in this slide, I've actually used a different representation of phage. This is what I call our Dalek representation, which I much prefer to the very long phages. This is like something out of Doctor Who, and we rather liked it, so we've been using this very simplified representation. So what we have here is the antibody genes on the inside of the phage, and the antibodies, the antibody fragments, so-called single-chain Fe fragments, where we have a linked heavy and light chain variable domain on the outside. They're fused to the phage through the gene 3 protein. So then we tested the recombinant phage for binding to the hen egg white lysozyme, which was the antibody corresponding to these V genes. We'd taken a monoclonal antibody, a hybridoma, and we'd cloned it in here, and we found out these phages, using an ELISA assay, bound very selectively and specifically to hen egg white lysozyme, but not, for example, to turkey lysozymes. They're very specific, and everything looked very good. So we now mixed the phage antibody with a large excess of wild-type phage and attempted to enrich the phage antibody to demonstrate the other work of George Smith and Parmalee, the affinity selection process. And we found that we could select this phage by multiple rounds of affinity columns. In fact, on one pass, we could enrich the phage, our antibody phage, 1,000-fold, and by growing the enriched phage and passing it down the column a second time, we can enrich it a further 1,000-fold, which means we've got 1,000,000-fold enrichment over 2 rounds of selection. And in principle, using 3 rounds of selection, you could get 1,000,000,000-fold, and then you could go up to George's gazillion if you just increase the number of cycles. So we were very pleased, really, that these folded fragments could express on phage, because it certainly wasn't clear to us that they would, but it turned out to be, the system seemed to work beautifully. So we then turned to making antibody expression libraries on phage. And the library was a so-called random combinatorial library. It was a type that had been described earlier by the Scripps group, in which a library of antibody heavy chains is combined directly with a library of light chains, usually from the same immunization. And this process, this process can be expected to generate the original heavy light chain combinations of the B cell that it originated from, as well as entirely new combinations. And this is shown on this slide. So let's take a very simple case of 2 B cells with encoding 2 different antibodies, one with a red and white light chain, and the other one with a red heavy chain and a white light chain, and the other one with a green heavy chain and a black light chain. So if we clone out those genes and we shuffle them all up, we end up getting 4 possible combinations, 2 of the original and 2 new ones, the red blacks and the green whites. Now, as you increase the number of B cells that you use, you decrease the probability of finding the original combination, and you increase the probability of finding new combinations. So, for example, if you took 1,000 B cells, in principle, which were all different, in principle, you'd get 1,000,000 different combinations by combining them, of which 1,000 would be the originals and 999,000 would be new combos. So we were very interested in new combos because actually, if we were to take antibody genes from humans, we knew we needed to generate new combinations of heavy and light to generate new binding activities in order to be able to find things in there that would bind to the self-antigens. We knew the immune system would have deleted all of those original combinations that bound to the self-antigen. So for our first library, we took the V genes from the spleen of a mouse immunized with a hapten, phenyloxazolone. Now, you may wonder what a hapten is. It's essentially just a chemical entity that's coupled to a protein. And it's been used by immunologists for many years to study how the immune system works. And it simplifies the understanding of how the immune system works and the kind of antibodies that you get to different targets. Now, from a library of about 1 million clones, we obtained phage binders with a range of different heavy and light chain pairs. And we couldn't distinguish the original from the new ones, but some had binding affinities comparable to the very best hybridomas. In other words, in the order of 10 nanomolar. These were hybridomas made by Cesar Milstein from that same spleen, because we ran the thing in parallel. We cut the spleen in half, he did one bit, we did the other. The affinities he could get were as good as the ones that we could get, which was a great irritation to him. But the technology was looking very promising. With our second library, we faced up to the altogether more difficult problem of making human monoclonal antibodies without immunization. After actually quite a few technical improvements I don't have time to go into, we created a larger and more diverse library, this time from the white cells of human blood donors. From a library of 10 million phage clones, so this is tenfold bigger than the one I've described earlier from the immune mouse, we managed to isolate binders to many different antigens, foreign antigens, human self-antigens, but these had binding affinities in the micromolar range, and those affinities are too poor to use for therapeutic use. So we knew we'd got to improve those affinities, so I'm just going to focus on the left-hand side of the slide. We decided to improve those affinities. Let's follow what the immune system does. So the immune system takes selected B-cell clones and it subjects them to random mutation and further rounds of selection with antigen. So, for example, we took a phenyloxazolone binding phage antibody from our human library with a rather modest micromolar affinity, and we grew it in a bacterial mutator strain. And after multiple rounds of growth and selection, increasingly stringent selection, we managed to get mutants crawling out at the end with affinities 100-fold greater, now in the low nanomolar range, which is quite comparable to the affinities of mouse monoclonal antibodies made by repeated immunizations of the same antigen in a mouse. We could even construct a genealogical tree, and we've got some attempt to show this here, in which you could see the sequences that by sequencing all the mutants that we got at different rounds, we could identify 4 sequentially acquired mutations, each of which was responsible for lifting up the affinity and giving us this great increase in binding affinity. The right-hand side of the slide describes a shuffling approach, which I'm not going to go into, but it's an alternative and perhaps simpler way of creating, faster way of creating affinity improvements. But it's not enough, or it wasn't enough, just simply to create mutations. We also needed to find ways of selecting between those phages with different binding affinities. And to do this, we applied increasingly stringent affinity selection. So, for example, using longer wash times, so all the weak binders washed off and the strong binders stayed stuck to the support. The other possibility was to use low concentrations of antigen. So here, what I've shown here is on the left, is we've biotinylated the antigen, let it equilibrate with the phage, and then captured the phage bound to the antigen using streptavidin beads. And that also showed that we could apply that and fish out selectively those phages with the higher binding activity. But there was a confounding issue, which is touched on here, and that is that we discovered, and we hadn't really expected this, whereas in George's case, that the phage generally, all the antibody heads express a peptide, but in the case of the antibodies, we found that a lot of our antibody heads were chewed off. So a phage antibody might have between 1 and 5 heads, each species actually presenting different binding avidities to a solid support, which greatly complicated our selection of those with the highest binding affinity. So we reduced this problem by an approach devised by Jim Wells, in which what he did was, I think he'd done it for a slightly different purpose, in which we created what's so-called monomeric phage with fewer antibody heads. So actually what we end up doing is infecting, or sorry, we encode the antibody fusion on a so-called phage mid, and we use a helper phage to supply the other gene 3 subunits. So we end up with antibodies which have a single antibody on there, or otherwise they're completely bald. Now the bald ones don't bind to anything, so that's very easy. So you just end up with those which are bearing the antibody. So as well as developing libraries of V genes from these B lymphocytes, we also had been exploring the creation of synthetic V gene libraries by a combination of the underlying V gene segment building blocks. Now, the advantage of synthetic antibodies is that the diversity of the library is known, and we can tune it to order. But at that time, the sequences of most of the human V genes were not known. And if we wanted to use this as building blocks, we had to find them. So we started mapping and systematically cloning and sequencing all of the human antibody building blocks. And then we assembled them to create our largest and most diverse phage library of more than 10 billion clones, including almost all of the human V gene segments. So we've now got a huge library, and we could select that. So we assembled the library, these V gene segments as indicated on the left, and then we selected that library and against foreign and self-antigens. And we now found antibodies with binding affinities in the nanomolar range, quite comparable to the affinities of hybridomas. In fact, really, by using these very large libraries, we avoided the need to do so much affinity maturation. So the great thing about this is, from a single very large library, you could fish out antibodies to all kinds of different targets. Now, in parallel with our work at the MRC, the startup company, Cambridge Antibody, was working with commercial partners to develop human therapeutic antibodies. Their most spectacular success was the development of the phage antibody adalimumab, which is otherwise known as Humira, which is targeted against TNF-alpha, an inflammatory mediator, and it's used for treatment of autoimmune inflammatory diseases such as rheumatoid arthritis, psoriasis, or Crohn's disease. Now, when we started the work on adalimumab, the random combinatorial libraries of human antibodies just weren't large or diverse enough. We couldn't find anything in there that would be useful. So we had to try a different strategy, and we decided we would try and bootstrap, starting with a mouse antibody. So we have a mouse antibody, which I've marked in red, and we took this, for example, here, the mouse heavy chain variable region. We paired it with a library of human light chains. So you end up with this hybrid, half human, half mouse. We're selecting on antigen, and we did in fact find we got some binders by doing that. And now what we did was we combined that selected human chain here with its partner chains from a human library, and essentially selected again. So in a couple of steps, we'd managed to create a human antibody essentially from a mouse template. So I wouldn't call it a humanized antibody. There's no trace of mouse in there, other than we have built this human antibody using this mouse template. So it's essentially rather like the sort of grandfather's axe business, the handle, that my grandfather changed the head and my father changed the handle, and it's my grandfather's axe. Well, it's completely different. The whole thing is completely different. It's a different entity altogether. Now, other than adalimumab, Cambridge Antibody technology and later MedImmune, into which it was incorporated, are focused on creating very large libraries, usually not synthetic, but from human donors, of about 100 billion clones. And they end up getting high affinity antibodies from those. And I'm grateful to Jane Osborne of MedImmune who used to work in Cambridge Antibody before they were taken over, for the information on this slide. So the upper portion shows the range of pharmaceutical target classes, including targets here, which MedImmune has made phage antibodies to. And you can see a whole range of growth factors, chemokines, ion channels, receptors, GPCRs, cytokines, protease inhibitors, and peptides. And on the lower portion of this slide, down here, you can see those phage antibodies that are already on the market. So they've gone through the whole process of clinical trials and testing. And there are 60 more still in clinical trial, still moving through the system. And so, in conclusion, you can see that this technology of these antibody repertoires with the filamentous phage has actually proved extremely powerful way of harnessing evolution and is delivering a great new wave of antibody medicines. With that, thank you.
Frances Arnold was born in 1956 in Pittsburgh, United States. She got her PhD in 1985 at the University of California at Berkeley. She is now active as a professor at California Institute of Technology in Pasadena, also in California. Frances Arnold pioneered directed evolution of enzymes. She used her engineering skills to make this method a most versatile tool for developing biocatalysts for new environments and for new tasks. She has made a strong effort to make this planet a better place. The enzymes she develops can be used to speed up chemical reactions in a sustainable way, making less waste in a cleaner process. We call them fantastic biocatalysts. Now, Frances Arnold, I invite you to the stage to deliver your talk.
Thank you. I'd like to thank the Royal Swedish Academy of Sciences, the Nobel Prize Committee in Chemistry, my family, many of whom are here, my friends, even more of whom are here, and especially I'd like to thank my former and current students, and I know there's a group of you here, because it's really their contributions that I'll be talking about today. I'm an engineer. I have been in awe and mesmerized by the chemistry of the biological world for some time, because it's this incredible diversity of transformations that gives rise to all the beauty and functionality that we see in the natural world, the complexity of the products that nature makes. And when you think about how she can assemble these products with ease and elegance and remarkable efficiency from simple, renewable, abundant starting materials. To me, that's a real inspiration for chemistry of the future. Now, who does this chemistry? It's the enzymes, these remarkable, marvelous molecular machines that catalyze all the reactions in the living world, almost all the reactions in the living world, and that are DNA encoded. Now, I'm also in awe and inspired by the enzymes but also by the process that nature uses to create them. And in fact, that nature uses to create everything in the living world, evolution. It's a remarkable diversity-generating machine. It's a simple algorithm of mutation and natural selection that gives you a recipe for adaptation, for optimization, for innovation, and has created all the life that we see around us, starting from a universal common ancestor, a simple life form, more than 3 billion years ago. Now, I wanted to build new enzymes, and protein engineers, like I was in the 1980s, we struggled mightily with a simple problem, and that is we didn't understand, and we still don't today, enough about how a DNA sequence encodes function in order to be able to reliably design a new enzyme. That's why I turned to nature's process. So here's the problem. Even today, we can read DNA. We can write DNA. You can just email a sequence off to your favorite supplier, and you'll get it back in the mail. We can edit DNA, CRISPR-Cas. We have all these tools, but what we do not know how to do, and this is the problem that I took on 30 years ago, is how do you compose it? It's like a Beethoven symphony, the code of life. It's intricate, it's beautiful, but we don't know how to write like that. But nature does. You have this process. So we can think of all the proteins that exist today, in fact, all the way back to the universal common ancestor, as this set of proteins that are relevant to biology. And biologists, this is what you study, this little green place, because that is but an infinitesimal fraction of the possible proteins. We can think of this universe of all possible proteins as a playground for the future. This is where I want to explore, and I want to explore here because I think the solutions to many of humanity's biggest problems lie in this space. Now, this is a challenge, of course, because this space is vast. It's bigger than the universe, the number of particles in the universe, by many, many orders of magnitude. This remarkable short story by Jorge Luis Borges, The Library of Babel, describes, for all practical purposes, an infinite library of all possible books. And the librarians there are in despair. How could I, if I want to search this universe of sequences, which is like all possible proteins, but I know that most of those possible books encode nothing at all. They're just gibberish. These random combinations of letters don't compete with pumping oil out of the ground, for example. How could I search this library and find something meaningful? So this is a tough problem because if you look at this space, it's so big. And we've heard some very big numbers in these experiments. Gazillions. Well, I cannot search for gazillions. I don't have 10 to the gazillion graduate students or even 10 to the 9th graduate students. And if we look at, for a large protein, now we've gone from peptides to antibodies, which can be quite large, but we go to something like an enzyme, that space of possibilities is enormous. If you make a single mutation to a typical protein, you have thousands of ways to do that. You start making 2 and you have millions of ways. You make 3 and it just grows exponentially. We cannot search this space just without thinking a little bit about the process. Now, John Maynard Smith helped me address this problem in a marvelous paper that he wrote, published in 1970, my favorite 1.5-page paper. And he said, consider proteins in an ordered space where every sequence is surrounded by its neighbors that differ by one mutation. And he argued that if evolution is to occur, and he believes it does, and I think most of us here believe that evolution happens, Functional proteins have to be distributed in this space so that any functional protein, something you can scrape off your shoe, is surrounded by at least a few other functional proteins. Forming a network may be sparse in the space where most sequences are gibberish, forming a network where evolution can walk along. And that's exactly what proteins have done for the last 3.5 billion years. They've accumulated mutations, one at a time primarily, to create what the proteins are today. Now, of course, I want to make better proteins. I want to make enzymes that will solve human problems. So let me add one more dimension to this, and that is the fitness, the performance, how an enzyme performs a new job. And that's defined by me in this set of experiments. And we can think of this now as an optimization problem on a fitness landscape. Now, in the 1980s, we knew very, very little about the structure of this landscape. It could be rugged. And if it's very rugged, then most mutations are deleterious and send you into a crevasse of non-function. And evolution is very hard in that case. On the other hand, if it's smooth, and John Maynard Smith helped argue that it has to be smooth in its dimensions for us to be here to talk about it, then you can use simple optimization processes. Now, science, like all human endeavors, is an evolutionary process, right? It builds on what is there, and we recombine different pieces from different places. And a lot of interesting things were happening in the 1980s. You've heard about some of those. And in the early 1990s. For example, Manfred Eigen was talking about experimental molecular evolution. And Jerry Joyce was evolving RNA in the laboratory, directing the evolution of RNA. Jeremy Knowles showed us that beneficial mutations in proteins could be accumulated. Stuart Kaufman was talking about the theoretical fitness landscape searches. And then for me, Dan Dennett, a philosopher, gave me wonderful words and framework for the ideas that I was trying to work on. And all of these came together in a simple process. Now, the difference between my experiments and those that you've heard about is that I don't have an affinity selection that lets me search through 1 million or 10 million or 1 billion different molecules. We have to measure them one by one, because you get what you screen for in this business. And if you're looking at an enzyme, you need to measure what that enzyme does. Maybe we can test a few thousand things or a few tens of thousands of things. Most of those mutations are deleterious. So this meant that we had to be very careful about how we were going to carry out this optimization, making just a few mutations at a time, maybe one at a time randomly in the molecule, screen carefully to measure the small effects that you would expect to get from a single mutation. And then if you wanted something that was very different from what you started with, and believe me, choosing that starting point is important because you don't get a racehorse from a donkey. So you choose that, that's a very rational thing. If you want something quite different from what you start with, then you have to iterate. You have to accumulate those mutations over multiple generations. And this gave us an experimental process that has proven its value. So if you start with your donkey, and most enzymes are pretty good. Nature has done a good job optimizing them for their job. But if you start asking it to do a new job, for me, they're less enthusiastic, let's say. And that fitness drops dramatically. But on this smooth fitness landscape, a simple optimization of accumulating mutations over multiple generations can re-optimize it for the new job. That work from the 1980s that we started this culminated in this first paper published in 1993, where Kevin Chen showed that we could adapt an enzyme to what was considered a highly non-natural environment. In fact, it was so non-natural that many biochemists told me, oh, you can't do that. It won't work. An enzyme will never work in dimethylformamide. In fact, they don't like it at all. But what we showed is that with a few mutations And doing this process, you could convince it to work just as well in 50 or 60% DMF as it did in its natural environment. In 1995, I met Pim Stemmer, shortly after his DNA shuffling paper was published. We met at a workshop at the Santa Fe Institute, and I was taken with his vision. He introduced this idea of sexual recombination. So this is another search strategy for evolution. Seems to work pretty well. There's a lot of diversity you get by recombining your DNA with somebody else's. And Pim talked about this as a molecular breeding process, and I often use that when I present my work. Now, sadly, Pim passed away in 2013, but he started a company that pushed a lot of this technology, mine and his, out into the world. And as a result of their work at Maxigen and many, many other laboratories, this directed evolution process has created enzymes that you will find in your laundry detergents, that are used to manufacture pharmaceuticals, that are used in all sorts of different industries today. I'm very proud of those applications, none of which I'm responsible for. But that's the nice thing if you provide a basic framework that's simple. And this is what engineers like to do. Make it as simple as possible, then the whole world will be able to use it. But we learned some very interesting things about enzymes in the process of doing that. Of course, they will adapt rapidly. We didn't know this at the time. We didn't know how many iterations, how many mutations it would take to get a new function out of an enzyme, or to make it function in a non-natural environment, or take a new substrate. And those came rapidly. We also learned that we're a long ways away from being able to design them, because I knew this process would be useful back in the 90s because we knew it was fast, but also that the solutions that you got were so surprising. Mutations happened all over the protein, 20, 30 angstroms away from the active site, where no one could explain them, much less predict them. So here we have this marvelous optimization mechanism. And that's what I've told you about. You take a biological enzyme and you can optimize it and change it for different functions. So now I'm going to fast forward into today so that I can end with the future. I'm interested today not in the optimization so much, although that's very important, but actually how does evolution create novelty? How could you create enzymes to catalyze reactions you cannot even find in the biological world, or at least we haven't found them yet? You cannot go to a database and find an enzyme that catalyzes your favorite reaction. How could we import reactions even invented by human beings, 100 years of creative chemistry? How could we import that into the efficiency of the biological world and genetically encode whole new chemistry? And at first I thought that would be a really hard problem, because we all know enzymes are so beautifully designed. They position their functional groups to catalyze a reaction and stabilize a transition state. And how do you create a whole new active site? It's a very conservative process, what I just described to you, making one mutation at a time. But that's the wrong way to think about it, because nature's making new enzymes all the time, usually to break down your billion-dollar antibiotic, but usually in response to pressures that humans have placed on this planet. So here's a great example. A potent herbicide that we've been dumping into the environment, onto our fields for more than 60 years, was first thought to be non-biodegradable. Then all of a sudden, some enzyme figured out how to dechlorinate atrazine, giving a rich nitrogen source to the organism that invented it, and it spread all over the world. This is evolution in real time. Evolution in real time, every time you use antibacterial soap. We're causing and pushing evolution, but it's happening quickly. So how does this happen? How do you get evolution in real time? Well, it happens that enzymes already have those capabilities. They're like graduate students. You're selected for working long hours at low pay, and you can do that, but maybe you could also play the piano. or do dishes if you had to. If chemistry was no longer a viable field for work, you could go and switch into something else. Well, enzymes are just like that. Any enzyme that exists today has promiscuous capabilities that evolution uses as a fuel for further invention. And if you can find those promiscuous capabilities, I'm the trainer of molecules, I have to see those, then you can use this simple turn the crank, presto results process of directed evolution. So let me give you an example. I love the cytochrome Cs because here's a family of enzymes that nature has diversified to carry out all sorts of interesting chemical reactions, and many of you are familiar with these. Nature uses one or a set of closely related, highly reactive intermediates to do this. And presumably through this process of mutation and selection has diversified this family. Now we argued in looking at this that, wow, this looks a little bit like human invented chemistry. If you could convince a cytochrome P450 to create a reactive intermediate that has a carbon, these are reactive carbines in the active site, make an iron carbanoid. Maybe you could convince it to transfer that and do human chemistry like turn double bonds into cyclopropanes or insert that carbene into an XH bond. This is not known in the biological world. It was invented by human beings. But if we provided the right synthetic environment, think atrazine, right, a new niche for the enzyme, and this tool of directed evolution, perhaps we could create enzymes for these new human things. So it becomes a question, can a heme protein create these new reactive intermediates? And there was some evidence that that would be possible. But then the hard part, would it take that, instead of using it to chew itself up, would it transfer that to a second substrate? So now you've got to have two substrates coming together in an enzyme that's never seen these before. And if you can see that little bit of activity, does it evolve? 5 years ago, we asked these questions, and I'll just give you one example of much more recent work that we did. In fact, these do carbene transfer. We've created a whole bunch of carbene transferases, and one of the ones that I love to talk about is one that does chemistry that even humans have a really hard time doing, making highly strained carbocycles like bicyclobutanes, which is a transformation not known in the biological world. And even in the chemical world, the world of human chemistry, is very difficult to do, and it requires precious metals. So iron, earth-abundant iron, was never known to do this. But Kai Chen showed that he could make cyclopropenes. We have more than 50 kcals per mole of ring strain in these structures. And that by providing a carbene precursor to this, the enzyme would form the reactive carbene, transfer it to that, and that furthermore, it evolves. So Kai Chen discovered an enzyme sitting in our refrigerator. I have a tremendous collection of interesting heme proteins, and he just said, can you do this as a promiscuous activity? And then he showed he could evolve it over multiple generations to become a reasonably efficient catalyst. But even more wondrous is that he could further train the enzyme to do it twice. So if it hangs on to this reactive cyclopropene in the active site, he can now add a second carbene. So you blow off nitrogen with this, that drives the formation of the carbene. You can now add a second and make bicyclobutanes, pure diastereomers. You only make one of the 4 possible diastereomers because the enzyme creates the environment where this remarkable chemistry can take place. Furthermore, the enzymes can make lots of bicyclobutanes. Now, as far as I know, nobody wants bicyclobutanes. If anybody wants them, please let me know. But these are interesting molecules that really have evaded human chemistry. But here an enzyme just learned how to do that in a few generations. And they happen inside living cells. So you do this chemistry just by providing the substrates to bacteria in a culture, and they'll spew out enough of these so that we can assign the absolute stereochemistry. Let me give you another example. Consider bonds that are unknown in biology. Here we have silicon an element that is so abundant on our Earth's crust and has never been found bonded to carbon in the biological world. There are no carbon-silicon bonds in biology. Some people have wondered why that's the case. I'm not going to answer that question, but now they are found in biology because if you go to my refrigerator, you can find proteins, for example, from this hot, salty pool in Iceland, from an organism that grew there. And that protein, if you just search through these, that protein will take a carbene precursor and this silane, dimethylphenylsilane, and insert the carbene into that silicon-hydrogen bond and make new organosilicon compounds. Just a wild-type protein. No one ever asked if nature could do it. Of course, these are not naturally abundant compounds, but nature's capable. Nature has the inherent capability to do organosilicon chemistry. 40 turnovers, that's as good as the best human-made catalyst. Just a natural protein, 97% enantioselectivity. Oh, and by the way, I just point out, this is a cytochrome C, and it is so stable you can boil it. Even a chemist can use it. It's so stable, it just looks like a powder, and it works beautifully for this chemistry. The native function of this cytochrome C is electron transfer, and in fact, if you know anything about cytochrome C, you'll be puzzling, whoa, how does this happen? Because the active site where this chemistry takes place, if you look at the crystal structure of this protein, you see that the active site is all filled up with protein. The iron is fully saturated. All the ligands from the porphyrin ring and the axial ligands above and below just fill up the active site. So if you roll your little ball over the computer, you find a big fat 0 for active site volume. But nature doesn't care about your calculations because this protein is perfectly happy to do this chemistry. And our pictures, our simple pictures of proteins don't do justice of what's actually happened. Not only does it do this chemistry, but this protein also evolves. So that if you make mutations near the active site over 3 generations, you can evolve the low promiscuous activity of the wild protein into something that now becomes dozens of times better than the best human catalyst. Just turn the crank, give them a little bit of sugar, and the proteins will evolve. And it works inside of bacterial cells. Why is that important? The bacterial cells will make your catalyst and make millions of versions of your catalyst for the price of sugar. So if it's DNA encoded, and these are all just, you put the gene into the bacteria, if it's DNA encoded, you can just grow all the catalysts that you want and mutate it 1,000,000 times for the price of sugar. And here I show you an example where, for example, you could choose between putting that carbene on silicon or on nitrogen, and if you've evolved the catalyst, it chooses only the silicon. So you get chemoselectivity, enantioselectivity, all done by the bacteria. Now, when this story was published, it went all over the world because everybody said we're making silicon-based life. That wasn't the case. I don't have time to go into some of the funny things that came about it, but apparently nobody actually read the paper. They only read the title of the paper. So it went around the world and mutated the story as it went around the world. And it gave my group a lot of fun, laughter, reading the Twitterverse interpretations of how we were creating silicon-based life. But it works in life, and you could conceivably build silicon into living systems now that you have genetically encoded chemistry. You can also learn how the protein learned how to do this. You could get the structure of the protein, and in this case, we see that the structure has changed dramatically. We have a loop over this active iron that has completely flipped out into solvent. There were 3 mutations that were made in this protein. It's caused this loop to flip out and opens up a volume, a pocket here that has sufficient volume now to accommodate a large number of different substrates and do this chemistry extremely efficiently. Dozens of times better than the best catalyst that was ever published by a human being. If you can do it with silicon, you can do it with other atoms as well. And so, for example, recently, a year later, we just published, excuse me, the incorporation of carbons into boron H bonds. And yes, there are no boron-carbon bonds found in the biological world. And here's a bond that's very much favored by chemists, a very versatile system, organoborons. But you can take an enzyme or a protein like cytochrome C and just turn the crank. So you're starting to get a little bit of the pattern here. You discover a little bit of activity, you have this wonderful optimization process, and we can find by starting with the 0 generation wild-type protein and evolving over 3 generations, you now have something that when you use it under optimal conditions is 400 times better than the best human catalyst for the same reaction. Does it with earth-abundant iron. And you can, of course, also change the selectivity, tuning the catalyst for the chemistry that you're interested in. I'll just point out that in the biological world, one criticism we face if we want to do biocatalysis is that, oh, you can't do what I can do. I have great chemists at Caltech who can do and create materials that biological world can't do. But I hope that's going to change, because I've just shown you, even though biology is limited to the bonds that it can make. So carbon, for example, is often found bonded to nitrogen or oxygen or sulfur, a little bit to the halogens, a few other things, lots of hydrogen, of course, but there's vast portions of the periodic table that we have never found a biological system to do. And I just showed you in the course of 2 years, we've added 2 whole elements to biology's repertoire. So you can now build carbon, boron, and carbon silicon scaffolds in living systems. And of course, then do further chemistry on that in metabolic pathways. So I would like to leave you with this idea for the future, because I'm really excited about now, but even more so about the future of this science, that we have this incredibly powerful design algorithm, right? It works at all scales, from molecules to ecosystems. And it not only optimizes, but it can innovate, and it can do it in real time. And in the chemistry world, what that does is open this opportunity basically to take down the barrier between the natural chemistry and human-invented chemistry, and just import all those human inventions into the biological world. And just sit there, and I sit there and read chemistry papers. My students read old Jack's journals, and they say, wow, I bet I could import this reaction in. And I haven't shown you many of the other ones that they've been successful in doing. But this is an exciting thing, I think, for the young people, because by marrying these different fields, it's like you're in a gold mine of possibilities that you can now genetically encode and make much easier some of your favorite chemistry. And I'd also like to leave you with this idea that the biological world is wonderfully diverse. There is a bazillion proteins out there just waiting to solve your problem. And nature knew this, right? The internet of things. No, it's the internet of living things that's been crowdsourcing, problem solving for more than 3 billion years. And if we learn how to use this to our benefit, we can move into a much more sustainable future and make our fuels and chemicals using biological systems. So I'd like to leave you with this idea that we have a universe to explore. We have a spaceship for exploring that universe. It's called evolution. And I want to thank the very large number of people who've worked with me over the years at Caltech, big fraction of them are shown in this picture. They came for one of my big birthdays a couple of years ago. And once again, I thank them, and I thank you for your attention.
Francis Arnold, George Smith, and Gregory Winter, thank you all for wonderful talks. And I now welcome all three of you up on the stage to take all the applauses. Okay.